Welcome. Today we would be understanding the excretory system in human beings as well as lower animals. We would be understanding the structure of kidney, the structure of nephron, and how actually the concentration of filtrate takes place, and finally the disorders associated with excretory system. So those would be the major topics that would be covered for today. Excretion can simply be in three forms, that is, it could be excreted as ammonia, urea or uric acid. So when I say ammonia, that means it is the most toxic form. It is usually seen in lower order organisms where the excretion takes place either by diffusion or by skin or by gills. So those are the methods through which actually excretion takes place, and the product of excretion is ammonia. Now this is usually seen in the amphibians, the aquatic animals and the bony fishes. However, in higher order organisms, the form of excretion changes into urea, and this urea is relatively less toxic. So it is seen in mammals, terrestrial amphibians and marine fishes. So the important difference is here we have marine fishes where ureotelic excretion occurs, that is excretion as urea. However, in bony fishes we have excretion as ammonia, and therefore it is known as aminotelic. And the last form is the most concentrated form of excretion, which is the uric acid, and it is in the form of small pellets usually seen in reptiles, birds, insects and snails. So those are the common creatures under which excretion occurs as uric acid, and since it occurs as uric acid, it is known as uricotelic. So we understood three forms of excretion broadly, which is ammonia, urea and uric acid. Ammonia is aminotelic, urea is ureotelic and uric acid is uricotelic. So there is the difference between the three. Ammonia is the most toxic form usually seen in aquatic animals, bony fishes and amphibians. Ureotelic, I repeat again, is seen in mammals, terrestrial amphibians and marine fishes, and uric acid is seen in reptiles, birds, insects and land snails. Understanding the various excretory organs. So in various orders of animals there are different types of excretory organs. In lower organisms we have protonephridia, or which are also known as flame cells. These are found in platyhelminthes, which are flatworms or planaria. Then there is annelids and amphioxus where you have the flame cells or protonephridia that is seen. Now let's say here we have a body of planaria or flatworm. Here you can see the tubules that are there, and these tubules, which can be zoomed out here, have the flame cells, and this is the further structure of the flame cells, and this is an example of protonephridia. So protonephridia, I repeat again, is seen in platyhelminthes. A good example is planaria or flatworm, as we can see here. The next is nephridia. Nephridia is seen in earthworm and annelids. So nephridia is seen in earthworms, and the structure of earthworm we have seen here. Now in this structure of earthworm you can see the cross section where you have the nephridium that is there, and this is the section where you have the nephridium. So nephridia, the plural of nephridium, is seen in earthworms and annelids. Now coming on to Malpighian tubules. Malpighian tubules is seen in cockroaches, and then we have the example of Malpighian tubule here. So this is the structure of a cockroach, and towards the midgut and the hindgut there is the Malpighian tubules that is present. Now what moves into the tubule is salt, uric acid and water, and what is excreted out is in the form of uric acid. You also have recycling of water and salt that occurs in the Malpighian tubule. So these are the major divisions of the Malpighian tubule and the major segments under which we have the excretory organ as Malpighian tubules in cockroaches. The next is very interesting, which is known as antennal glands, and since it's present towards the antennal side, it is also called as green gland because of its color green. As you can see here in prawn, what happens is there is green gland for excretion, and this green gland has a bladder with a tubule, and then there is an end sac where ultrafiltration takes place. The tubule is responsible for selective reabsorption and secretion, similar to the nephron which we would study in kidneys. And finally you have kidneys, which is seen in mammals and higher order organisms. Now kidneys we would understand in detail how the actual processing and filtration takes place through kidneys. So to repeat again, 5 major excretory organs that are unique and different. So we have protonephridia seen in platyhelminthes or flatworms or amphioxus. Then we have nephridia, which is seen in earthworm and annelids, Malpighian tubule seen in cockroaches, green gland and antennal gland seen in prawn, and finally kidneys in higher order organisms like mammals. Understanding the structure of kidneys. Now we have the kidneys that are found in mammals, as we have seen. Now this kidney, the structure itself is interesting. It is a bean shaped structure which is very small. You cannot imagine, it's just 10 to 12 centimeters in length. Then you have a width of merely 5 to 7 centimeters, a thickness of only 2 to 3 centimeters, and weight of only 120 to 170 grams, and still is able to filter nearly 180 liters per day, of which the concentrated urine that is finally released is just 1.5 liters. So you can understand nearly 99% of it is reabsorbed through the kidneys. We would understand how it actually takes place in a while. Now, first of all, the location of kidneys. Kidneys are located in the last thoracic and the third lumbar region. When we talk about kidneys, there is the kidneys occur in pair, and then these kidneys are attached to ureter, and finally this ureter enters or moves to the urinary bladder, where you have towards the end the urinary sphincter, which is controlled by the CNS or central nervous system, finally through which you have the excretion of urine that takes place. Now this kidney itself is made up of millions and millions of nephrons. The role of the nephron is very similar to what we have seen in lungs, the alveoli. Alveoli are responsible for exchange of oxygen and carbon dioxide. Similarly, so basically filtration or purification of blood at that stage, and here nephrons are again responsible for elimination of nitrogenous waste from the body. So that is the basic idea that we see. Now coming on to the structure of kidney, the internal structure of kidney. So as we said, kidney occurs in pair. We would understand just a single kidney here and understand the internal structure. So first of all, there is an outer layer which is known as capsule. Within the capsule, there is the inner layer. Now inner layer is further subdivided into outer region, which is the cortex, and the inner region, which is the medulla. So that is the division of the kidney itself. Now the point where kidney connects to ureter is known as helium. Now this helium, this is not the gas helium, this is Hi, not He, which is the element that is found as helium. So this helium has 3 important components. What are those 3 important components? There is renal vein, renal artery, and renal nerve. So all these 3 components pass through this helium region. So you have the renal vein, the renal artery, this is the ureter, and then you have a renal nerve that pass through this region. So this notch, which is known as the helium. Then there is the calyx. Now this region, as you can see here, is the calyx. Now calyx could also be divided as major and minor. The center region, as you can see here, closer to the pelvis. So this is the region of the pelvis, and closer to the pelvis, so this is pelvis, and closer to the pelvis there is major calyx, and towards the periphery, as you can see here, this is the minor calyx. So calyx is further seen, the major and the minor calyx we have understood. Now this cortex further extends to the medulla, and there are certain gaps that are seen. Now these are known as renal columns or the column of Bertini. So this is the column of Bertini, and now within this kidney structure you have millions of nephrons. Now interestingly, nephrons are present in both the section, the cortex as well as the medulla. We would understand in a while how there is a division of the nephron through the cortex and the medulla. So let's understand the structure of neuron first to understand which region is present in cortex and which region is present in medulla of the kidney. So coming on to the structure of nephron. As we said, within one kidney there are millions and millions of nephrons that are present. The nephron itself is an interesting structure that we would understand. So towards the beginning of the nephron there is a Bowman's capsule fitted with glomerulus. Glomerulus is nothing but a tough network of capillaries which includes the afferent arteriole entering into and the efferent arterioles moving outside. So this is the afferent arteriole that moves in the glomerulus, and then there is the efferent arteriole that moves out of the glomerulus, and this efferent arteriole further moves and we can say curves itself around the loop of Henle within the nephron, and this curve is what is known as vasa recta. So very important, what is vasa recta? So vasa recta is the capillary which is surrounding the loop of Henle. Now together, Bowman's capsule and glomerulus, which is the initial part of the nephron, is known as the renal corpuscles. Now, renal corpuscles move horizontally. This section where it moves horizontally is known as PCT or proximal convoluted tubules, followed by the loop of Henle. So this is the structure of the loop of Henle. In the loop of Henle, there is one section that moves down, and since it's moved down, it is known as the descending limb. The other section moves up. It is a U-shaped structure. The section that moves up is the ascending limb. So two limbs of loop of Henle, the descending limb as well as the ascending limb. And finally, this loop of Henle moves to the next section, which is the DCT or the distal convoluted tubule, and this distal convoluted tubule finally connects to the collecting duct, and that is how we understand the basic structure of nephron. Now in the previous section we said there is certain portion that comes in the cortex of a kidney and certain portion that comes in the medulla of the kidney. So let's understand the Malpighian corpuscle, which is the renal corpuscle, along with the PCT and the DCT, the proximal convoluted and the distal convoluted, comes in the cortex region. Only the loop of Henle is present in the medulla of the kidney. So what I can do is I can simply divide a line here, and with this division of line I can say the region which is north of it or above it, we could say, is present in the cortex region. The remaining region is present in the medulla of the kidney. So what is present in the cortex? To repeat again, it is the Bowman's capsule and the glomerulus, together known as renal corpuscles, along with the proximal and the distal convoluted tubules. The section of loop of Henle surrounded by the capillaries vasa recta is present in the medulla. Interesting to note again, if the loop of Henle is small, it extends very less into the region of medulla, and this section is known as cortical nephrons. However, in some cases, the same loop of Henle is very long and enters deep into the medulla. When it enters deep into the medulla, it is known as juxtamedullary nephron. So two types of nephrons again to remember. One is where the loop of Henle is small, and this is known as cortical nephron. The other where the loop of Henle is long, extends deep into the medulla of the kidney, and this is known as juxtamedullary nephron. A very interesting concept to understand again. As we said, glomerulus has a tuft of capillaries. You have the afferent arteriole that enters and the efferent arteriole that moves out. This efferent, which moves away from the glomerulus, forms a network around the renal tubule, and towards the region of loop of Henle, as we said, it is known as vasa recta. But the whole of the capillaries that surrounds the structure of the nephron is known as the peritubular capillaries. So the name is peritubular capillaries. And the region of the peritubular capillaries closer to the loop of Henle is known as vasa recta. Now vasa recta can be, just remember, it can be absent or reduced in cortical nephrons but not in juxtamedullary nephrons. So that is one of the basic understandings of the structure of kidney. To summarize again, kidney occurs in pairs. It is bean shaped in structure. The internal structure of kidney we understood, which starts with the ileum, where you have the renal vein, artery and nerve that enters in, and then enters in or moves out, and then you have the capsule. Within the capsule there is the inner part. This inner part has cortex as well as medulla, and then each of the kidney has millions of nephrons, and each of the nephron has a renal corpuscle which is composed of the Bowman's capsule. Along with the Bowman's capsule there is glomerulus. This, along with the proximal distal tubule and the proximal and the distal convoluted tubule, that is the PCT and the DCT forms, is present in the cortex of the kidney. The region of loop of Henle is present in the medulla. If the loop of Henle is small, it is seen in cortical nephrons. If it is long, it is seen as juxtamedullary nephron. This is about the kidney and the structure of the kidney. Let's understand the process of urine formation and concentration of filtrate, which is one of the most essential parts of the nephrons that is there. Now, understanding the formation of urine. First of all, urine formation occurs through three stages. First is filtration. The second is reabsorption. And the third important stage is secretion. Let's understand the three stages one by one. The first stage, which is the stage of filtration, occurs in the glomerulus and the renal corpuscle. Now here what happens is nearly 1100 to 1200 milliliters is filtered per minute, and of this filtered blood, which is 1/5 of the blood that is being pumped by the ventricles of the heart, is there. Now this process occurs in three layers. What are those three layers? First is the endothelium of the glomerular blood cells. The next is the epithelium of the Bowman's capsule. Now this epithelium of the Bowman's capsule is also known as podocytes. From these podocytes, you have minute spaces which are known as filtration silt, through which the process of filtration takes place. And the third is the basement member, which is attached between the two, which is the endothelium of the glomerulus and the epithelium of the Bowman's capsule. Between that, you have the basement layer. So all of these three are responsible for the process of filtration. This process requires a lot of reduction in the filtrate, and therefore this is also known as ultrafiltration. So where does ultrafiltration takes place? The ultrafiltration takes place in the glomerulus region, and the blood is filtered so finely that all of it except proteins pass to the Bowman's capsule. So the only thing that passes from the blood to the Bowman's capsule at this stage is proteins. Now, the glomerular filtration rate is around 125 milliliters per minute. That means nearly 180 liters of blood is being filtered on a daily basis. So a very high capacity that is seen for the glomerulus, which is present in each of the nephron. So we have a structure of nephron, and there are millions of such nephrons present within the kidney. Now, towards the distal convoluted tubule and the region where you have the efferent arterioles, there is the JGC, which is the juxtaglomerulus apparatus, and this juxtaglomerulus apparatus is important sensitive region, which is, as we said, the modification of the distal convoluted tubule and the efferent arteriole system. Once there is the fall in the glomerulus filtration rate, which is the GFR, what happens is the cells of the juxtaglomerulus region activates, and this leads to stimulation of blood flow in the glomerulus region. So there is a stimulation of the blood flow in the glomerulus region, and finally it brings back the filtration rate to normal for the glomerulus filtration rate. Okay, so the next work that we see is reabsorption. Now where does reabsorption takes place? There is 180 liters of blood that is filtered on a daily basis by glomerulus. However, the urine that passes is just 1.5 liters. So there is lots and lots of reabsorption that occurs. This reabsorption occurs through the renal tubules. Now, what are the things that actively reabsorb and what are the things that passively reabsorb? So in the case of passive reabsorption, we have only the nitrogenous waste that is there. However, when I say there is active reabsorption, this includes reabsorption of glucose, it includes reabsorption of ammonic acid, it includes reabsorption of sodium ions, so ammonic acid, then you have sodium ions that are reabsorbed in the case of active reabsorption. Passive reabsorption only for nitrogenous waste, and then water is also reabsorbed passively only in the initial segment of the nephron. So those are the reabsorption process that occurs. However, there is an important process which is secretion. Now secretion is the tubular cells actually secrete hydrogen ions, potassium ions and ammonia into the filtrate that is there. This is secreted by, the reason for this secretion is to maintain the ionic balance and the acid-base balance of the body fluid. So in order to maintain the acid-base balance and the ionic balance of the body fluid, there is secretion of hydrogen ion, potassium ions and ammonia into the filtrate in the region of DCT or the distillated convoluted tubules, the distal convoluted tubules. So there are three processes, I repeat again, in urine formation. The first is filtration, which occurs in glomerulus, and after this happens, it is passed into the PCT. There is reabsorption in the renal tubule, which is the PCT as well as the loop of Henle, and finally there is secretion to maintain the ionic and the acid-base balance in the DCT, and this is where you have the ions of hydrogen, potassium and ammonia that is released into the filtrate. So this is the process. Now, within the components of the nephron, what is the role of each of those? So let's first talk about PCT or proximal convoluted tubule. Now proximal convoluted tubule is composed of simple cuboidal brush border epithelium that is present. The idea is to increase the surface area for absorption. Nearly 70 to 80% of the electrolytes and water is reabsorbed in the PCT itself. So 70 to 80% reabsorption occurs in the PCT for the electrolytes. Water is also reabsorbed. It helps to maintain the pH and the ionic balance by secreting hydrogen ions, ammonia and potassium into the filtrate. As we have seen, the hydrogen ion, potassium ion and ammonia into the filtrate. Now as we saw this process in the previous slide, the secretion occurs in the DCT mainly, but it also occurs in the PCT here. And as we said, PCT is responsible for most of the electrolyte reabsorption Also, the brush border epithelium helps to increase the surface area for reabsorption. The next is the loop of Henle. We have the descending limb and the ascending limb. Now, what is the difference? The descending limb basically is permeable to water. That means, as you can see, the water can move out here, but it does not allow the moving of the electrolytes. However, in the ascending limb, it is impermeable to water, but it is permeable to the transport of the electrolytes, as we can see. So, sodium ion, chloride ion can move out of the ascending limb of the loop of Henle. So, descending limb and ascending limb, very important. Reabsorption is minimum in the descending limb because of the high osmolarity of the interstitial tissues. However, it increases as you move towards the ascending limb. So, reabsorption is technically more in the ascending limb as compared to the descending limb of the loop of Henle. Finally, you have the DCT or the distal convoluted tubule. It is here where there is conditional reabsorption of sodium ion that occurs. Along with that, there is reabsorption of carbonates and there is selective secretion of, again I repeat, hydrogen, potassium and ammonia into the filtrate in order to maintain the ionic and the acid-base balance of the body fluid. Now, finally it connects with the collecting duct. Collecting duct is seen in both the regions. So, you have the region of cortex as well as the medulla where you have the collecting duct. So, there is part of collecting duct in the cortex and the remaining part of collecting duct which is seen in the medulla. However, the PCT, DCT and the renal capsule is seen only in the cortex. Loop of Henle is seen in the medulla region. Okay, so collecting duct is where most of the water is now reabsorbed and finally urine is released, which passes into a small amount of urea as well to keep the osmolarity of the to maintain the osmolarity along with the pH balance. And finally there is the selective secretion of hydrogen and potassium ions that again takes place here. Now, understanding the concentration of the filtrate is very important. Kidney produces urine which is 4 times much more concentrated than the initial filtrate that comes in. Now, how this process of concentration actually takes place? This process of concentration occurs through a counter current mechanism. Now, how this counter current mechanism takes place is very important to understand. This is the structure of nephron. This nephron is surrounded by the capillaries which are known as vasa recta, as we have studied before. Now, vasa recta is the capillaries that surround the nephron and specifically the loop of Henle. Now, what happens is there is flow of filtrate on the 2 limbs of loop of Henle and how they differ. So, understand, I am drawing 2 U-shaped diagrams here. One diagram is the loop of Henle, the other diagram is the nephron. So, this is the nephron and the next is the loop of the vasa recta which surrounds the loop of Henle. So, within the nephron there is the loop of Henle and surrounding that there is the capillaries which is the vasa recta. How it happens? Now, the filtrate moves through the loop of Henle in the direction that we are drawing. Okay, so it's just in the direction that we are drawing, and in the descending limb, as we said, it is permeable to water. However, the ascending limb is impermeable to water but permeable to electrolytes. So sodium and chloride would move out. However, the reverse happens in vasa recta which surrounds this loop of Henle. So there is a loop of Henle surrounded by the capillaries, and in this capillaries the movement occurs in a reverse direction as the loop of Henle. So just simply the opposite direction. And here what happens in the ascending limb of the vasa recta, not the loop of Henle, what happens is in the ascending limb you have the 2 limbs here, and here it is permeable to sodium and chloride. However, the ascending limb of loop of Henle was impermeable to water, and here it is impermeable to the sodium and the chloride ions, and the descending limb here is basically where you have the removal of the sodium and the chloride, that is, it is permeable to sodium and chloride. So what is happening in the nephron, as simple as that, the loop of Henle, the reverse happens in the vasa recta. So therefore it goes up, and from the capillaries it goes down, and therefore there is a counter current mechanism that operates within the region of loop of Henle. This helps to increase also the osmolarity. So in the cortex region the osmolarity is around 300. However, in the inner medulla it increases to around 1200. And this is where the sodium chloride is transported in the ascending limb of the loop of Henle, which is exchanged by the descending limb of the vasa recta, and this helps to maintain a counter current. So to have a simple understanding again, this is the loop of Henle which is inside, and surrounding this loop of Henle there is the vasa recta outside. So what happens is, within the loop of Henle the things go in this direction, and within the outer region, which is the vasa recta, the things start to flow in the opposite direction, and this creates a counter current mechanism. And with this counter current mechanism, the concentration gradient of the interstitial medulla is being maintained. It helps in easy passage of water and further concentration of urine to nearly 4 times the initial filtrate that was done by glomerulus. Now, this whole process of kidney functioning requires various organs. So first is regulation through hypothalamus, the next is juxtaglomerulus apparatus, and finally we have heart. So let's understand these one by one. Hypothalamus releases ADH, which is the antidiuretic hormone, which is responsible for vasopressin secretion. So vasopressin secretion takes place through the hypothalamus. Now what happens by the ADH? ADH facilitates water reabsorption in the later part of the tubule and therefore prevents the diuresis to occur. However, if the body fluid increases, the ADH would decrease, as simple as that. Now, ADH also affects the functioning of the kidney because it helps in as a constrictor effect of the blood vessels that takes place, therefore increasing the blood pressure in the glomerulus filtration rate and the glomerulus blood flow. So that is the role of hypothalamus. The next is the GJA, which is the juxtaglomerulus apparatus. Now here, the fall in the glomerulus filtration rate would activate the juxtaglomerulus apparatus or the GJ cells to release renin. This renin would release angiotensinogen 1, and then you would have angiotensinogen 2 that would be released. Angiotensinogen 2 is a vasoconstrictor. So it increases again the glomerular blood pressure and activates the cortex to release aldosterone. Aldosterone now causes reabsorption of sodium and water from the distal part of the tubule and leads to increase in the blood pressure of the glomerular filtration rate. And basically, this whole complex that is formed is known as a renin-angiotensinogen mechanism. Okay, so angiotensin mechanism, so renin-angiotensin mechanism is being formed. To repeat again, juxtaglomerulus apparatus, there is secretion of renin. Now this renin releases or helps to convert angiotensinogen in the blood to angiotensinogen 1 and finally angiotensinogen 2. Angiotensinogen 2 is a powerful vasoconstrictor, so it increases the blood pressure in the glomerular filtration rate or the glomerular blood flow and therefore creates a complex which is known as renin-angiotensinogen mechanism. The next is the role of heart. So through the atria of the heart you have ANF which is released, which is the anti-natriuretic fluid. Now this basically is a vasodilator. So far we were talking about vasoconstrictors, which were constricting the blood flow and increasing the blood pressure. However, ANF is a vasodilator and it decreases the blood pressure in the glomerular blood flow and therefore checks the renin-angiotensinogen mechanism that is being formed. So that is the basic idea how the regulation of kidney functions takes place. Now to summarize again, there is three processes which is involved. First is filtration, reabsorption and secretion. Filtration starts with the glomerulus. Nearly a lot of filtration occurs in that region and nearly 90% of the reabsorption occurs through the loop of Henle, the ascending and the descending limb. And finally, there is secretion of hydrogen ion, potassium ion and ammonia in the PCT and the DCT. Then there is concentration of urine that takes place with the help of counter current mechanism that operates between the loop of Henle and the vasa recta. So the flow of blood is reverse in the case of loop of Henle and the vasa recta, and this creates a counter current mechanism which increases the concentration of urine to 4 times the initial filtrate, and this whole process is being regulated by hypothalamus, GJA, which is the juxtaglomerulus apparatus, and heart. Heart releases ANF, which acts as vasodilator, reducing the blood pressure of the glomerular blood flow and checks the renin-angiotensin mechanism that is being formed. However, hypothalamus releases ADH or vasopressin and the juxtaglomerular apparatus releases angiotensinogen 2. Both of those are vasoconstrictors. That means they increase the blood pressure in the glomerular blood flow and the glomerulus filtration rate. So this was about how actually the urine formation and concentration of filtrate occurs and the various organs responsible for the constriction and the dilation. Let's understand the process of micturition, other excretory organs and disorders. Now, the first thing we understand is micturition. So, micturition is the release of urine from the body, and this occurs through the control system, which is through the nervous system and the central nervous system. Since I say it is controlled, that means it is voluntary in action and occurs through the urinary sphincter. Now, the urine that is released has a distinct odor, has a pH around 6.0, that means it is acidic, and urine which is around 1.5 liters per day of excretion that takes place, of which around 25 to 30 grams of urea is present as the byproduct. So this is the various studies that we see here. Now coming on to other excretory organs which are important to understand. So within the other excretory organs besides kidney, there are lungs, skin and sweat glands. So when we talk about lungs, the lungs filter the carbon dioxide and provide us fresh oxygen. When it comes to kidney, it filters and eliminates the nitrogenous waste. One of the very interesting findings you would know is about saliva. Saliva also has a capability to eliminate little of nitrogenous waste. However, if the kidneys do not function properly, there are various disorders associated to those, which we would understand in a while. And let's say a person is suffering from diabetes mellitus. In that case, within the kidneys, there would be presence of glucose and ketones in urea. When there is glucose in urea, we call it glycosourea. And when there is ketones in urea, we call it as ketonuria. Now, as we said, lungs as one of the excretory systems, excretory organs, then kidneys as one of the excretory organs. We also have liver as one of the excretory organs. Now, what does liver eliminate? Liver secretes bilirubin, biliverdin, cholesterol, and then you have steroids, vitamins and drugs. Then you have the skin. Through the skin, we have the sweat glands. Now, this sweat glands releases water along with sodium chloride, urea in small proportion, and then lactic acid, specifically when there is lot of exercise that takes place. This helps to cool the body and remove the waste from the body. Also with the skin, we have the sebaceous glands. Now these sebaceous glands are the oil glands. They eliminate sterols, hydrocarbons, waxes through the sebum which is produced, which is the oily covering for the skin. So sebum is also eliminated. So those are the various eliminations that takes place from the body, the various organs associated with elimination. The most important is, as we said, kidneys, lungs, then you have liver, skin and sweat glands and the sebaceous glands that are seen. However, as we know, there are disorders associated with kidneys. Now the disorder is first of all understood if there is presence of urea in the blood. If there is presence of urea in the blood, this condition is known as uremia. This condition can lead to kidney failure, and there is the process of hemodialysis that takes place, which is an artificial kidney. Now how does it happen? A person is basically connected, the arteries are connected and the blood is drained from the body. Now when the blood is drained from the body, it is important that it should not coagulate. So anticoagulant agents like heparin is given to the patient, okay, and then this heparin, with the help of this heparin, the blood is drained. Now this blood which is drained enters into a selectively permeable membrane where there is dialyzing fluid or dialyzing solution that surrounds the blood that moves in. Now important to note that this blood, this dialyzing fluid does not have nitrogenous waste. Since it does not have nitrogenous waste, it allows the blood to get clear, and finally the cleared blood is moved through the vein back into the body of the patient by adding anti-heparin agents. So first we added the heparin so that there is no coagulation of the blood and the blood is easily able to flow through the hemodialysis system, and finally when it is to be entered into the body of patient, again anti-heparin is given so that the process of normal blood coagulation that occurs in human body can take place. However, in extreme cases, if there is a kidney failure, the only way is transplantation. Now, transplantation can be accepted from a donor which is one of the closest relative in order to reduce or minimize the chances of immune system rejection, and therefore kidney transplants, which is a highly complicated process, is most successful with close relatives. So that is one of the disorders. We call it condition of uremia, which in severe cases can lead to kidney failure and in extreme cases could require transplantation. Then we have the next important disorder associated with kidneys, which is common and called as the renal calculi. Renal calculi implies there is presence of stones or calculus in the kidney, and these calculus are nothing but oxalates. The last important disorder that we would understand is glomerulonephritis. Glomerulonephritis is the term itis, wherever it is connected, it talks about inflammation. So there is inflammation of glomeruli within the nephrons in the kidney. As we have understood the structure of kidney, kidney is composed of millions of nephrons, and each nephron has a renal capsule, which is the glomerulus fitted within the Bowman's capsule, and this glomerulus, when inflamed, is a condition which is known as glomerulonephritis. So those are some of the major disorders that we have understood associated with excretory system. We would be covering many more interesting lectures for you. Have a good day.